Hello, everybody. Welcome back. We are on to Chapter 6, Energy Flow in the Life of a Cell. So last lecture was covering Chapter 5, which was Plasma Membrane Structure and Function, talking about the phospholipid bilayer and concentration gradients and how things move across the plasma membrane. We will be seeing some of those ideas again in a couple chapters, chapters 7 and 8, photosynthesis and cellular respiration. So you can kind of think of chapter 5 as almost foundational ideas, setting us up for the processes that happen inside of cells later on. You can also think of chapter 6 as kind of the same way. We are going to be presenting some of this foundational information about energy, about how it's used in the cell, and how introduce this idea of enzymes and what they do. So if you're reading through chapter 6 and you're like, I kind of get it, but I don't really see where it fits, that's okay to feel that way, because we are going to apply this information in chapters 7 and 8 when we get into those processes of photosynthesis and cellular respiration. So just kind of keep that in mind as you're working through this material. It will all kind of come together in those later chapters. We'll see that. Okay, so I have my introductory picture here showing some marathon runners running across a bridge compared to some race cars going around a track. So my question is, how are these the same? So what's going on between these living organisms and these mechanical machines? Well, the answer is they're both using energy. They're both utilizing energy, converting energy from one form to the next. And they're actually pretty similar in that we use the energy stored in food as chemical energy, and we convert that into mechanical energy of the act of running. So, contracting your muscles, breathing in and out, pumping blood around your body. Whereas the cars do pretty much the same thing. They harness the chemical energy that's stored in the gasoline and convert that to the mechanical energy of pumping pistons, spinning the crankshaft, and moving the gears to make the car go. So humans running or moving and cars racing around a track actually are very similar in regards to utilizing energy and conversions of energy. Okay, so since this chapter is going to be dealing with energy, we need to answer the question, what is it? So what is energy? Well, the kind of textbook definition is energy is the capacity to do any kind of work, which seems pretty simple. But what does work mean in the science context, right? So work, you go to work, I got to go work out, but that's not quite the same thing. So in science, work is defined as a force acting on an object that causes the object to move. So like this guy here in the picture, it looks like he's doing some kind of weight competition where he's moving this huge tire. So he is using his energy to move the tire. That movement is called work. So we're going to be taking a look at energy a lot in this chapter. So when we are thinking about that, all this discussions about energy, we need energy to do work. Now that could be pumping our blood, contracting our muscles like he's doing, or it could be at the cellular level of pumping ions. So if you remember in active transport back in chapter 5, we use ATP, we use energy to pump calcium against its concentration gradient. We're still moving stuff, that is work, and we have to use cellular energy in the form of ATP to do that. All right, so we've just talked about energy, so now we get to take a look at the types of energy. I'm going to go to the pendulum here real quick because it's going to stop this animation before I have a chance to talk about it. So our two types of energy are potential and kinetic. Potential is the stored energy, and kinetic is the energy of movement. So when you're taking a look at that pendulum swinging, when it is moving, you can see the kinetic energy bar goes the highest. When it reaches that peak, if it was to pause at those kind of high ends of the pendulum, then that's the potential energy. If we could like grab it and hold it, that would have potential energy. So right now it's stopped on one side, so we can see the potential energy is very high and the kinetic energy is 0 because it is not moving. That green bar represents total energy. So within this particular system of a pendulum, whenever it reaches that high, kind of the highest point in its swing, the total energy is stored in potential energy. As it was swinging, that movement, that is going to be the kinetic energy, and the potential energy is released into that kinetic energy. by the nature of doing these videos, I can't restart that animation. So hopefully you kind of saw it go back and forth to see that relationship. The other one here, this picture in the upper corner, this is another type of representation of these two types of energy. We have potential energy when the bicyclers reach the top of the hill, energy input into that bicycle to go against gravity. And so now he's at the top of the hill. He's got some energy stored in that gravity, if you will. And so then when he kicks over to the other side, he doesn't have to put in any more energy. He's releasing all of that stored potential energy as he drives down the hill. So we'll be seeing in the cell these two types of the energy, potential energy and kinetic energy. Okay, so let's take a look at how some of this potential energy can be stored. And one of the main ways that we're going to be taking a look at that is chemical energy. It's a very important type of potential energy in cells. So let's take a look at some examples, and you are probably familiar with this. You just may not know it's called potential chemical energy. So the food that you eat. So the chemical bonds in the macromolecules, we got to go back to chapter 3 a little bit. So carbohydrates and starches, lipids, proteins, nucleic acids, all of those were really big molecules, and there's energy stored in those covalent bonds. And as it is stored in the food, this is our potential energy. So we can utilize that to drive our cellular processes. That's what we're going to get to in chapters 7 and 8 when we take a look at photosynthesis and cellular respiration. Gasoline. We had the examples of the runners and the cars earlier. So the potential energy for the runners to power themselves through their marathon is food, and the potential chemical energy that the cars use is in gasoline. So you can see the chemical formula. It's just carbons and hydrogens. It's called a hydrocarbon. And so that is a type of potential energy stored in those chemical bonds. When you have a fire, when the wood has, it's basically starch, it's cellulose. It is a large polysaccharide with a ton of energy stored into it. It is when you burn a fire, you are releasing that chemical energy, and you then are producing heat, radiant energy, which is a different type that we're not going to spend too much time on. But the cellulose that's stored in the wood is a type of chemical energy. And then lastly, you probably have these around your house, batteries. There's chemicals inside batteries, and there's a potential energy. And so as you use your batteries, you are converting that potential energy into kinetic energy if it's causing things to move physically, or if it's just moving electrons through the circuitry of your electronic device. Okay, so that was kind of our foundation of energy. Now, when you study energy, you study what's called thermodynamics. So we have a couple kind of universal laws that we have to lay down our foundation to understand a little bit about energy before we can start applying it to cellular processes. So there's two laws of thermodynamics that we're going to be taking a look at. So they kind of help to describe the availability and usefulness of energy. So the first one we're going to be discussing is the first law of thermodynamics. That makes sense. So it basically states energy can neither be created nor destroyed. So we just can't get something for nothing, basically. So when you have a given system, the total amount of energy will remain the same. We can't destroy it. We can't add more in that particular system just from nothing. It can come in from another system or remove from the system, but it's not going to be destroyed. You can't actually ever destroy energy. You can move it and convert it and change it, but you're not ever going to destroy it or create energy from nothing. So the example that they give in the book is gasoline. So let's say the gasoline tank holds 100 units of chemical energy. They're trying to use some easy numbers for math. So if the gasoline can provide 100 units of energy, you put that in your car, you let it do what it does, you explode it, it releases that potential chemical energy in those bonds, uses it to drive the car. The result is it may seem like we've destroyed that gasoline, the energy that's in that gasoline, but really we've just converted it to different forms of energy. So combustion engines are not very efficient. So for that 100 units of energy that's stored in the gasoline, only 20 units of that energy is converted to kinetic energy to actually move the car. 80 units of that energy is lost as heat to the system. So it's not very good energy. It's a kind of random, chaotic energy. It's not really usable form of energy, where the 20 units is the kinetic energy, which you can use. So internal combustion engines are only about 20 to 30% efficient for all of this energy that's stored in the gasoline. Most of it is lost as heat. That's why we have to have radiators to cool down our motors, or else, we'd have a big problem with our cars not running correctly. All right, so that was the first law. Energy can neither be created nor destroyed. The second law of thermodynamics has to do with the organization of energy. So there is a word that's associated with the second law called entropy. So entropy is a fun kind of a thought and a concept, if you will. So it is the measurement or the kind of the disorder of a system, the randomness, the chaos, the measurement of that randomness of energy is what we call entropy. So in the second law of thermodynamics, as reactions take place, as energy transfer happens, entropy increases. So what that means is disorder or chaos or randomness increases during these processes of reactions. So I know my head is kind of cutting off some of those words there, so you can look it up on the PowerPoint file itself. Basically what this means is if we do not keep putting in energy to maintain our organization, right, because living things are complex and organized, what happens is things start falling apart. So your cells cannot maintain homeostasis, your organs cannot maintain homeostasis, and ultimately your cells die and your body dies when you cannot input enough energy to fight against that entropy, if you will. So entropy is always wanting to happen. Things always go from order to disorder. If we want to maintain order, we have to put in energy to do that. All right, so here's kind of some visual representations of this idea of entropy. So in that the top part of that, we have a couple boxes of little blue dots. So it says, if the particles represent gas molecules at normal temperatures inside a closed container, right, so that's the box, which of the illustration configurations came first? So do you think if I were to write a time arrow, would that go in, let me see if I can get my pen here, would we be going in this direction? So all of the gas molecules that are spread around, they're going to come together after a period of time, or will we be going in this direction, where all of the concentrated gas molecules are going to spread out? Well, you should know the answer to that based on information we learned in chapter 5 when we were talking about diffusion, where things like to go from an area of high concentration to low concentration. So that whole discussion that we had back in chapter 5 kind of follows this second law of thermodynamics. Things like to go from order to disorder. So if we were to take a look at the actual arrow, the time arrow, so as time goes by, the high concentration of gas molecules is going to evenly disperse. And evenly dispersed, randomly moving around gas molecules would be an increase in entropy. We went away from order towards disorder. Okay? The second example in that picture is if you had a whole bunch of bricks and you threw them off the back of the truck, which pattern of order would most likely happen? You throw them off the back of the truck, would they stack neatly in a nice little pile, or would they be all randomly kind of piled onto the ground? Well, I'm sure you could answer that question. Disorder is the more probable outcome for that particular example. You may have done this yourself. Everything's nice and neatly stacked. Maybe you go and haul wood. It's all nice and neatly stacked in the truck. You dump it out. It's not going to turn into a nice, neatly stacked pile of wood on your driveway. It's going to be a big mess. And then you have to put in energy to go stack it, elsewhere, in your backyard or something if you use firewood for your heating source. So those are some real world examples of how entropy increases. Here's another one that you may be more familiar with, is your bedroom, right? Or your kids' room. So this is entropy in action. Disorder happens spontaneously. If you were to not input any energy in your living space, it would quickly become more chaotic and random and disordered. And if we wanted to go back to a more organized state, you have to put in energy. So organization requires energy. So when we start talking about cellular processes like photosynthesis and cellular respiration, these things we are going to have to fight against entropy for a lot of those because we want to maintain a homeostasis And to do that, things have to have order and patterns, not entropy. So entropy is not good for cells to get stuff done. Okay, so the majority, like 98.9% of life on this planet, again, there's a few exceptions of the deep sea hydrothermal vent communities, but with that exception in mind, everything on this planet requires energy from the sun. So that's our driving force. This is kind of what allows life to take place. So we have to take in that energy. If you are a photosynthetic organism, you have the ability to turn that sun's energy directly into energy usable in the cell. Are your autotrophs, right? So photosynthetic organisms. If you are non-photosynthetic, those are our heterotrophs, you have to consume energy in the form of the food that was made by those plants or animals, kind of if you think like a food chain, to be able to have that energy to maintain your homeostasis. So ultimately, all of our energy needed to do what we have to do to maintain order and homeostasis is derived from the sun. Don't you wish all kids would act like that when they're eating their vegetables? Okay, so as we are setting the stage for these processes, which have a lot of chemical reactions in them, let's talk a little bit about what a chemical reaction is. Some of you may not be familiar with that. We didn't really talk about chemical reactions too much in our chemistry chapters. So a chemical reaction is where chemical bonds are made or broken. So we are familiar with chemical bonds, right? Covalent bonds, ionic bonds for the most part, hydrogen bonds to a lesser degree. So in a chemical reaction, typically you are going to take reactants, which we can see on the left-hand side of this reaction arrow, and we are going to convert those to products. Now, the products are the end results of a chemical reaction. That could be taking a large molecule and breaking apart, like in our hydrolysis reactions when we take a large starch and we break it into smaller monosaccharides. Or it could be the dehydration synthesis, where we are taking smaller subunits and hooking them together into larger products of polymers. So you could have chemical reactions go both ways. They could be breaking things down reactions or building things up reactions. So we're going to be seeing some of these real life examples in photosynthesis and cellular respiration. Okay, so when we take a look at these different types of reactions, we can classify them or categorize them. More vocab words here in biology. So depending on the energy that's either being used or given off by these reactions, we can call them exergonic or endergonic. So the kind of the roots there, exer means out, like external, and ender means internal, and gonic is a root that has to do with energy. So we'll take a look at exergonic reactions first. So exergonic reactions are ones where you carry out the reaction from your reactants to your products, and in that process of converting your reactants to your products, you will release some energy into the system. Okay, it could be potential energy, it could be kinetic energy, it could be chemical energy, it could be light energy, it could be heat energy. Doesn't matter. It could be any kind of energy. It's just being released by this process of this reaction taking place. All right, so for example, when we take in sugars like glucose and our cells break them down, the potential energy that's stored in those chemical bonds in the glucose molecule, when that is released, it gives us the smaller subunits of like water and carbon dioxide, but in the process, we harvest and use that energy. So breaking down glucose into its products is an exergonic reaction. So here in this diagram, we have two reactants, kind of these yellow circles. When they go through the reaction to become the blue circles, those are the reactants, we also get some energy as a byproduct, kind of that big starburst there. So that's our exergonic reaction. The other type is endergonic. So endergonic requires an input of energy. You have to have energy to go into the system to go from your reactants to your products. What that means is the products have a higher amount of energy than the reactants that started the reaction. So the example of an endergonic reaction would be photosynthesis. When a plant has the ability to turn glucose and water, which are very low in energy, and convert it to glucose, which is very high in energy. So when we get into our photosynthesis chapter next, that is one big, long endergonic reaction. When we get into cellular respiration, which is after that, that's going to be one big, long exergonic reaction. So they're going to be very closely related, and you'll hopefully be able to see these terms a little bit more. Okay, so those were two different types of reactions, whether you are releasing energy as an exergonic or needing energy as an endergonic reaction. But all reactions, whether we are talking exergonic or endergonic, all need some little push to kind of get things started. And that little push, kind of this, what's called the activation energy, is what's needed to get everything started. The reason why is for reactions to take place, the molecules in the reaction need to be moving at a sufficient speed. It's all about this molecular atomic movement. Molecules bash into each other. Remember, it's those valence electrons that are involved in bonding. So a lot of times those, negative will repel negative. So if you have two atoms coming together, they may not want to be next to each other necessarily because of those negative charges, but we have a little bit of a push that's going to bring them right close together, so they can do that either swapping of electrons in an ionic bond or the sharing of electrons in a covalent bond. So the example, again, that's kind of blocked off by half my head, is when you are starting a fire in the fireplace. You have your wood, it's full of potential energy, you have your, and it releases energy. It's definitely an exergonic reaction, but you just don't put the wood in the fireplace and say, go, start. You need a little bit of a boost to get that going. So that's your match and your kindling. You have to put some activation energy into the system to then start releasing that exergonic reaction of combustion. All right, so here's a picture of the picture of the, sorry, I'm distracted because I have my little friend here. She's walking all over my desk. Okay, so like we had the fire example with the wood, you can do the same thing with your toast and marshmallows. We all know marshmallows burn. It's an exergonic reaction. We see a release of heat and light energy, but the marshmallow is just not going to start on fire all by itself. You need a little bit of that thing to get it started, like the match or your campfire. So in a graphical representation, we have on the left-hand side of the axis here, right here, this is representing the amount of energy within the reaction, okay? So the marshmallow or whatever. So here, when we are higher up on this graph, we have a higher amount of the energy in the molecules. When we are down low, down here on that part of the graph, we are lower amount of energy. So let's say we start here. This is our start. Okay, these are the reactants, and this is our finish, all right, or end. So that's where we want to go. So if we take a look at where our reactions start, they are right here. Okay, so comparatively, they have a higher amount of energy within their molecules. And if we come down here to where we end, maybe we'll change a different color. So here's our start, and here's our end. We can see on that y-axis there that the reactants have more energy than the products. Now we know we cannot destroy energy thanks to that first law of thermodynamics. So where does the energy go? It is released into the system, into the environment, into the cell, into the organism, or whatever the system happens to be. So reactants have a higher amount of energy. Our products have a lower amount of energy. So this is an example of an exergonic reaction. But to get the whole process started, you can see we have to, handle or deal with this little hump of required energy input that has to take place even in an exergonic reaction. So this is what we call the activation energy. So that would be the match to light the marshmallow, or the lighter stick to match your fireplace. These are examples of this activation energy. And it's the same thing inside of your cells with these chemical reactions and processes that we're going to be taking a look at. Okay, so in our body, in our cells, we have a whole bunch of exergonic and endergonic reactions happening all over the place. But what's nice about our cells is we are pretty efficient in that if we have a reaction that's going to be exergonic, so we go from the reactants through the reaction to the products, and it's going to be giving off energy, we just don't let all of that energy go to waste. We use that energy released from an exergonic reaction to help drive and be the energy input into endergonic reactions. And these are what we call coupling reactions. So you have an exergonic coupled with an endergonic, and the exergonic is going to drive the endergonic. So we kind of get a 2-for-1 deal, if you will. So when the products or the energy released in the exergonic reaction fuels an endergonic reaction, this is our reaction coupling. So the example, the burning of gas in your car is an exergonic and energy releasing process. And we harness that to move the pistons, that potential to kinetic energy. Moving pistons is an endergonic reaction. You have to put in energy. So in your engine, we have coupled the release, that explosive release of energy in the combustion of gasoline to drive the energy needing requirements of moving the pistons, right? So exergonic drives endergonic. Okay, so I'm just kind of going from topic to topic. Remember, we're just kind of setting the groundwork. We're laying some foundational information down. So if it seems like I'm going all over the place, I probably am. All right, so the next topic is called, or discussing what we call energy carrier molecules. So these are molecules inside of our cells that actually do the direct work, right? They carry the energy to get the work done. And we said that energy for us is stored in the food that we eat, right? It's got a whole bunch of chemical energy stored in the lipids and the proteins and the carbohydrates that you're eating, but we can't take that banana, like in the example here, and shove it into our muscles and have it work. We have to have an intermediate molecule. So these are our energy carrier molecules. So we harvest the energy that's in the banana, mainly carbohydrates, right? It's kind of a starchy fruit. And so we convert that energy stored in the bonds of the carbohydrate, and we convert it to more usable forms for our cell processes. And so these are what we call energy carrier molecules. They're kind of an intermediate, and they're only happening within the cell. So the most common one that's used all over the place inside of our cells, you may have heard of it before, is called adenosine triphosphate, and we usually abbreviate that as ATP. So ATP, if you remember, is made in the mitochondria. That was one of its jobs, is the powerhouse of the cell, right? So ATP is our temporary but very useful energy carrier molecule. So it is the product of some of these exergonic reactions. So when we burn glucose, right, the glucose in the food that we eat, and we're releasing that energy, we use that energy to and convert that over into this cellular energy molecule called ATP. And if you take a look at it, here it is on the top picture, right here. This is our full molecule of ATP, and it is formed by taking what's called ADP, which stands for adenosine diphosphate, meaning it just has 2 phosphate groups hooked on. And then energy goes into this reaction to put that 3rd phosphate group on to give you an ATP. So now we've got this high energy molecule. Now we can use that inside of the cell to do work. And then the way that happens is when the ATP gets broken down, that 3rd phosphate group, an enzyme comes in and breaks that off. It releases that 3rd phosphate group. That is an exergonic reaction. It's going to release energy. And then this energy right here, this is what we can do in our cell. This allows us to do work. Okay, so this is a role of ATP. An analogy that I like to use for understanding kind of that why ATP is such a good energy molecule is, if you remember functional groups back in chapter 3, I believe, one of them was called a phosphate group, and we usually see that as just the P with a circle around it, right? This guy right here, it's our phosphate group. It is a phosphate with 4 oxygens or 3 oxygens, and it's bonded to your carbon. So we've got a whole bunch of oxygens sticking outside of this phosphate atom, and oxygens don't really like to be around each other. So, because they're kind of slightly negative, remember they have that polar covalent bond properties. And so if you're trying to squeeze, you have 2 oxygen or phosphate groups with all these surrounded by all these oxygens, and you're trying to squeeze a 3rd one on there, there's going to be a lot of repulsion. So imagine you have this spring, and you're trying to, and you push it in and you kind of squeeze the spring, and then you have a latch. So that latch that's hooking that energy that you put into this tightly coiled spring, that would be like that third phosphate group. Okay, and so then when you release that latch, like breaking off that third phosphate group, it boing, the spring kind of like pops open and it releases that stored energy, that potential energy. So whenever we cram that third phosphate group on there, think of that as like tightening up the spring or putting energy into the spring, this coil. And so when we can, it's got a lot of potential energy now stored in that third phosphate bond. And so when we break that off, we are releasing a whole bunch of that stored energy. And it goes back and forth. Our cells are constantly making ATP and breaking ATP. That's why we have to keep eating, because the energy in the food that we take in drives this production of this molecule ATP, which our cells require to be able to maintain homeostasis, to do all the stuff that we need to do inside of our bodies. Okay, so here is a picture kind of showing this coupling reaction of our endergonic and exergonic reactions with ATP, and some actual examples of some of these macromolecules or biological molecules. So over here on the left-hand side, this is our exergonic reaction of breaking down glucose. So they're showing you that glucose, again, here's kind of that high level of energy on that left-hand side of our graph, if you kind of think back a slide or two ago. So glucose has high energy, high potential energy stored in those bonds. That is our reactant. Here's our reaction arrow, okay? And here are the products. So when glucose breaks down, it breaks down into carbon dioxide and water with the release of energy. So this is our exergonic reaction because energy is a product, okay? And so the energy is in the form of ATP, okay? So we're producing ATP by harnessing the energy stripped off of that glucose molecule. Now let's say you're weightlifting, right? Getting some strong muscles. You need to make proteins to make more muscle because you're telling your body you need more because you're lifting heavier weights. And so your body needs to make proteins. It has to hook all of those individual amino acids together, that dehydration synthesis reaction we saw before, and that requires energy. You're going from disorder, individual random amino acids, to an ordered, very complex protein. We have to go against entropy. We have to put in energy to maintain and get to order in this organization. So here is our endergonic reaction, right? So endergonic reaction, we take individual amino acids with the energy of ATP coming into the system, right? So amino acids have a very low energy. So if we're gonna take a look at the graph over on the left, and proteins have a very high energy, we are knowing we have to put energy into the system to go from amino acids to proteins. And look at that relationship. It's a coupled reaction where the exergonic release of energy by breaking down glucose allows us to do the endergonic process of making proteins. So hopefully you're kind of getting that idea of how this ATP molecule is an energy intermediate, kind of a energy carrier molecule for our cells. Okay, you'll be seeing a couple other electron carriers or energy carriers, in this case, the electrons specifically. So ATPs are primary, what we call the energy currency of the cell, a lot of times is what it's called. These other molecules are carrying electrons. They serve a purpose in helping along the way in the process of energy production and utilization. So electrons are usually the things that are being transferred between molecules. That's where the energy is. Remember, they can go from different energy levels in their shells, in their energy shells, in their orbits. So two common electron carriers that you're going to be seeing, one's called nicotinamide adenine dinucleotide. You don't have to know that full name, but we're going to be seeing it as NAD+, and then its energized form called NADH when it's actually holding on to those electrons. The other one is called flavin adenine dinucleotide, but again, we're not going to ever call it those names. We're just going to use its abbreviation, FAD and FADH2. Now, if you'll notice between these two electron carriers, this guy right here and this guy right here, they are missing H's in the NAD+ and the FAD form. When they are the electron carriers, we'll see that they now have H's. Okay. When we start talking about electron carriers, we're going to see these more in cellular respiration, chapter 8. It's going to be the hydrogens that are going to be transferring and carrying these electrons to hook onto these larger molecules. Because if you remember, what's a hydrogen atom? It's a proton and it's an electron. So if the electrons in the hydrogen atom are energized, we can move that whole hydrogen with its energized electron onto these electron carriers, and then they can carry that energized electron from one place in the cell to the next to get to the next reaction center to utilize the energy in that electron. So I just found a little comic here with the mitochondria in the background and two little carriers that got their bags full of electrons. This is representing your NADH and FADH2. So just think of them as, sometimes I think of them as electron buckets. It's just something that's going to be carrying those electrons in the kind of in relationship with these hydrogen atoms from one place in the cell to the next. All right, so here's just kind of a schematic of the role that these electron carriers do inside of the cell. So here we have, again, the high to low energy for reactants and products. So let's say you have this first oval here. We're going to start right here. So we have some high energy reactants. That could be glucose, that could be lipids, that could be proteins, whatever. It's got a whole bunch of that stored potential energy in it. And then in the process of this exergonic reaction, it's going to have some low energy products. And so where is that energy? It's now we're going to focus it more and kind of define that energy as these electrons. So the NAD plus, right? So this energy, NAD plus, is going to be added to this high energy electron to produce the NADH along with this hydrogen. And then what we can do is we can then carry that energy in the NADH over to somewhere else that needs to go against, right? So this is an endergonic reaction. So here's some more reaction coupling. The low energy reactants needs to make high energy products. And so here is the high energy that's going to go into that product. So this would be like photosynthesis, or this could be cellular respiration, where we have our stripping the electrons away from glucose. NADH is temporarily holding on to them, and then we can use that to make our ATP by cramming that third phosphate group onto it. We'll see that actually is the case when we take a look at cellular respiration. Okay, so that's the first half of this chapter 6, dealing with energetics and thermodynamics and what's energy and all this kind of stuff. So that's those topics will be applied to more in chapters 7 and 8 for sure. The next half, so the kind of the second half of chapter 6, is dealing with these things called enzymes. You may have heard enzymes before. We've kind of briefly touched on them when we were talking about proteins, because enzymes are proteins. They are molecularly proteins. They're not carbohydrates, they're not lipids, they're not nucleic acids. They fall into that fourth category of macromolecules of proteins. So if you remember, proteins are big long chains of amino acids. They fold primary, secondary, tertiary, maybe quaternary, and that three-dimensional shape is very important to their function. That's true of all proteins, and especially true of enzymes. So what are they besides being proteins in molecular nature? Well, what they do is their job is to speed up chemical reactions. We call this to catalyze, to be a catalyst for something. So they are often called biological catalysts because they work in biological systems, primarily inside of cells. And what they do is they lower that activation energy. So here in this picture, we can see that same graph that we saw earlier with our reactants having a high amount of energy and our products having a low amount of energy. So this is an exergonic reaction. But remember, we had to overcome that activation energy. That's this arrow here. Okay. I'll get rid of those guys. So then, in the presence of an enzyme, take a look at what happens. So this blue line says, okay, we still have a little bit of we still have to trigger this process, but an enzyme is going to make it happen a lot faster because our obstacle that we have to overcome is a lot smaller. So that's the role of enzymes, is they reduce that hump that you have to get over to get the reaction going. So this, they do this very quickly. We're talking like millions of reactions per second they can catalyze in certain enzymes. So that's three-dimensional shape allows it to do that. And so we're going to be taking a little bit closer look at these structures of the enzymes and kind of what they do or how they do what they do. Okay, so in biological and biology books, microbiology books, anatomy books, a lot of times when the pictures represent enzymes, they look like big amorphous blobs. So the purple blob in this picture is an enzyme. But remember, it's not just a big blob. It is that big, long sequence of amino acids folded into these very specific shapes. So it's really complex to show the accuracy of a protein in pictures and illustrations. So they just kind of get these three-dimensional blob shapes. So they serve the purpose, but just kind of keep that in mind. Okay, so when we are taking a look at an enzyme, we've got the bulk of the structure of the enzyme. That's this big purple blob. But then we have a very specific region on the enzyme called the active site. Now the active site is like a little pocket, or think of like a puzzle piece or a lock and a key. So this little pocket has a very specific three-dimensional shape made up by the amino acids in their proper order, and it will match the three-dimensional molecular shape of the thing that it works on, which is what we call the substrate. Okay, so the substrate will fit into that active site, right? So here's our new word. Substrate is what the enzyme acts upon. The active site is where it does this acting upon, if you will. So in this case, we are doing a synthesis reaction. So the blue and the green substrates start off as separate reactants. They come into that active site. The enzyme kind of distorts and folds and kind of pushes those substrates together, and that's what's going to trigger the chemical reaction. And so there will be a bond that's made, and so now we get a new product, kind of the pink blob over there. So that could be the reaction to, or the product of this particular reaction. Blue plus green equals pink, right? So that's the chemical reaction. The enzyme allows that to happen, and it happens very, very quickly within the cell. If we wanted to put some exact names of chemicals to this, we could do, now I'm going to have to remember my disaccharides. Maltose sugar was one of our disaccharides. It is made up of glucose and glucose. So if you have glucose plus glucose, you have an enzyme that hooks that together to give you maltose sugar. Or we have fructose and glucose, I believe is sucrose is our disaccharide. So you have two monosaccharides, an enzyme hooks them together to give you your disaccharide. That's exactly what would be happening here in this picture. Or if we get two amino acids hooked together, then we would get a dipeptide. Two amino acids hooked together. If they're starting off individually, this enzyme would hook them together, generating that peptide bond. So this is an example of, putting things together, a synthesis reaction, but it could work the same way. You might have a different enzyme with a different active site to recognize that pink molecule now, and then reverse that reaction and break it into the two products of the blue and the green, okay? So that would be like sucrose broken down into glucose and fructose. So there would be a completely different enzyme because it would have to match that three-dimensional shape of the sucrose, not the glucose and fructose. So that active site is very specific to the functionality of the protein. Okay, so when we take a look at enzymes and reactions, so again, my head's kind of blocking off this diagram here. It's on page 96 in your book. But this just goes from intermediate F, enzyme number 6, goes to a purple blob called G as the end products. So the way that enzymes are involved in reactions, we just kind of saw the cycle of how an enzyme actually works, is it catalyzes many reactions, not usually just one at a time. It can catalyze a whole bunch of reactions, or many reactions are catalyzed by enzymes. I should say that a little bit better because it's usually a different enzyme for every reaction. So when we talk about all of the chemical reactions that take place in our body or in our cells, this is what we call metabolism. It's the sum of all the chemical reactions in a cell. Metabolism, you've probably heard that word before. So metabolism is a collection of these pathways called metabolic pathways, and every metabolic pathway is catalyzed by a collection of enzymes. So if we take a look at pathway 1, here we start with our first substrate, this pink ball A. So it's going to have a very specific shape, and its enzyme 1 will be a big blob, right? big blob, and it's going to have an active site that will match, let me see, do the best I can. It's like a circle with a flat line, and it's kind of like a circle. So what's going to happen is this A is going to go in and fit inside that active site and get converted to this next product, which is B. All right, so A is converted to B via enzyme number 1. Now, that could be the end of the reaction, but usually our cells are a lot more complex than that. So then B needs to get turned into C, and it has to have a completely different enzyme because B is a different molecule than A. So enzyme 2 is going to have its own three-dimensional shape with its own specific active site that is just specific and special for B. B will get turned into C. C will get turned into D via a different enzyme, number 3, and then D will get turned into E with another enzyme 4. So each one of those steps is going to be catalyzed by a different enzyme. Now, sometimes reaction pathways can split. So in this case, down here in pathway 2, if we had a different enzyme, different than enzyme 3, right? They're both working on the same substrate, but they might have a different product. C gets turned into F, and then F gets turned into G, which is behind my head, okay? So those are our metabolic reactions catalyzed by enzymes, and every reaction has to have its own specific enzyme because of that specificity of the active site. Okay, so what if you have a chemical reaction in your body that you don't want to continue? You're like, okay, we're done. We have all the product we need to turn that off, or we need to turn that on. Let's activate an enzyme, and because we need this particular product inside of the cell. So we have ways in our body to regulate enzymes. We can increase the concentration of enzyme, or we can increase the concentration of a substrate, and that would definitely increase the reaction happening. But concentrations aren't always the easiest to regulate inside of a cell. You have limited space. You might have diffusion of things moving away, and so you can't compartmentalize your enzymes or your substrates. So it's not always feasible to increase rate by increasing enzyme or substrate. So there's other ways that a cell can regulate the enzyme activity. So the two that we're going to take a look at here are controlling enzyme synthesis, which I don't have a picture for, but I'll explain it, and then competitive and non-competitive inhibition. I forgot my parenthesis. That's okay. So when we take a look at controlling enzyme synthesis, I had to do a little bit of foreshadowing here because we'll actually get to this process at the end of the term. But enzymes are proteins, right? We've established that. Proteins are the product of DNA transcription and translation. We call gene expression. So in your DNA, right? So DNA is housed in the nucleus within all of our cells. There are little strips of bits of information along kind of encoded within the DNA, and that is called a gene, and that particular gene codes for the instructions of a particular protein, just one protein. So for example, we'll do growth hormone. So growth hormone is a protein-based hormone. So there's a gene located on your chromosomes that if you read that gene and it goes and gets expressed, it will produce growth hormone. So what we can do, instead of just producing a ton of enzyme and just produce a ton of enzyme, we can kind of dial back how much growth hormone we produce, or increase the amount of hormone that we can produce at the genetic level, at the DNA level. We can turn on that gene and we can turn off that gene, and that's going to affect how much enzyme we actually have. The second category are using inhibitors. So inhibitors block the active site from being able to carry out its reaction, and there's a couple different ways that it can do that. So the first one is competitive. So let's say we have this purple blob of an enzyme, and the blue looks like a flask almost. This is the substrate, right? So we have this very specific shape here. This is the active site shape. And so our substrate goes into the active site, voila, you have your chemical reaction. Well, what if you have something that mimics the substrate? It looks like it, but it's not the substrate, and it's going to actually block the reaction from happening. First of all, it will block any of the actual substrate from landing, and it itself is not going to be able to be acted upon because it's not the correct substrate. So this yellow ball is what we call a competitive inhibitor or competitive inhibition, because the inhibitor, the thing that's stopping the reaction, is actually mimicking or is similar enough in shape that it's going to bind to the active site, and then the substrate can't. The other type is called non-competitive inhibition. So if we take a look at our purple blob of an enzyme, we can see over here, this is in that picture A, this site is called the non-competitive, also known as allosteric. You might hear that word somewhere. Non-competitive inhibitor. So it's just another binding site elsewhere on the protein, on the enzyme. So if you have this orange blob over here, this other inhibitor, it's not going to block the active site, but take a look at what it does to the active site. So when that non-competitive inhibitor binds to that non-competitive site, it changes the shape of the active site. It's now a V shape. Our little flask of a blue substrate can't bind no longer in that active site. So we've in essence stopped the ability of that enzyme to function. So our cells use these regulators, competitive and non-competitive inhibitors, to change the activity of the enzyme. Okay, so there are also some other things that can influence enzyme activity. These may or may not be used for the cell to regulate its own enzyme activity. It could a lot of times be accidental exposure or changes in environment that could be detrimental to the ability of an enzyme to function. So a lot of times drugs or poisons affect the enzyme's activity by doing that inhibition, the competitive inhibition. Sometimes they bind permanently to either the non-competitive or the active site, and so it can't ever work again. Some of these inhibitors can be reversible. So it binds for a little bit and then it pops off, and it binds for a little bit and it pops off. So these inhibitors can be reversible or irreversible, and a lot of times these happen to be drugs or poisons that you could put into your body purposefully or accidentally, or some kind of inadvertent exposure. So the example that I have here is a purposeful exposure of your body to a particular drug. In this case, an antibiotic in the family of sulfonylamides. So this pathway right here is the normal pathway the bacteria use to make their nucleotides, right? So nucleotides are your monomer of your DNA and RNA. So if you're a bacteria and you want to replicate, make more copies of yourself, you need to make a whole lot of DNA and RNA nucleotides. So you can copy your DNA and RNA, obviously. But if you're a pathogen, so let's say you're E. coli or Salmonella or some other bacteria that you have now invaded the host, that's you, the person, you have a bacterial infection. So what you want to do as the host, you feel yucky, you want to get rid of the bacteria. So how can we do that? Well, one of the targets of some antibiotic drugs is this particular pathway. So if we take a look at what normally happens, here's our substrate. It's called PABA, right? That's the molecular shape. It fits specifically into the active site of this purple blob enzyme, and it produces a product called dihydrofolate acid, which then used by another enzyme, you can see it's slightly different, producing what's called tetrahydrofolic acid, and another pathway goes on, and then that gets you your nucleotides. So you have your precursor molecules, PABA, a couple enzymatic pathways, and you get your nucleotides. Okay. Well, this type of drug here, we'll skip over to the sulfonylamides. This sulfa drugs, if we take a look at it, right? So take a look at the PABA right here in that yellow box. Okay, it's got a carbon, double bond to an oxygen, single bonded to an OH. This is a carboxylic acid group, a COOH, one of our functional groups we talked about back in chapter 3. Now take a look right here in that yellow box. It's a little bit different, right? So the green box is exactly the same. So it's molecularly similar, but instead of a carboxylic acid group hooked to that carbon, we have a sulfur and a nitrogen containing group. So that's kind of the nature of these sulfa drugs. So this is a competitive inhibitor. It binds to that first enzyme, preventing the production of dihydrofolic acid. So if you're a bacteria being exposed to sulfa drugs, you cannot make your antibiotics and you die, which is good for the host because now you've gotten rid of your bacterial infection. Now you might be saying, well, how come we don't die? Don't we need nucleotides too? Yes, we do, but we do not use this pathway. We do not do a PABA to nucleotide pathway, so we do not have that first enzyme that the sulfonylamides would affect. The middle pathway is a different type of drug. So this is called trimethoprim. It's a different type of antibiotic, and it blocks that second enzyme in the pathway. So if we take a look at it, trimethoprim is similar enough to the dihydrofolate acid that it competitively inhibits this second enzyme, and so you cannot continue on in that pathway. So there's some real-world examples of how drugs can be competitive inhibitors to these metabolic pathways. Okay, some other situations that can affect your enzyme functions are more of the environmental conditions, not so much the inhibitors themselves. But because of that three-dimensional shape, that is very important. And certain environmental conditions can change that three-dimensional shape. So if we look at the little comic that I have here, it says, "Welcome, enzyme school alumni. Poor Jerry, I told him working in non-ideal pH levels would denature him." Right, ha ha ha. Got to know a little bit about some biology to understand that joke. But you may have remembered from our chapter 3 when we were talking about proteins, and when they get exposed to certain conditions, that three-dimensional shape can get destroyed, and that's what we call denaturing. Because enzymes are proteins, they are susceptible to those same environmental changes. So most enzymes have a very narrow range, what we call the optimal conditions for that enzyme to do its job. This is typically in temperature and pH. So again, when an enzyme loses that three-dimensional shape, we call it denatured, and it lost its ability to function. So pH, so if you have too acidic or too basic environments, salt, if it's really salty, temperature too hot, you can denature the enzymes, or we have what are called coenzymes. We'll see that more in chapter 8. But let's take a closer look at temperature and pH and how it can affect the enzyme. So when we take a look at pH, this is the graph that we're going to go with. I don't have a picture for salts, but it can just, the salts themselves can affect the enzyme by changing that structure. Denaturing is all about changing structure. So we have a pH scale on the bottom, kind of, it doesn't go all the way up to 14, but if you remember, neutral was 7, right? pH 7 had the same amount of hydrogen ions and hydroxide ions. That was back from chapter 2. So we have in our stomach, right? Our stomach has some hydrochloric acid in it. It's pH of 2. And we have an enzyme that works great in pH of 2. It's called pepsin. Its job is to break those peptide bonds and break down large polypeptides into smaller polypeptides and individual amino acids, eventually for intestines to be able to absorb that. Now, if you take a look at its curve, it doesn't work very good at pH of 0. And it doesn't really work very good at pH of, looks like 3.8. It works best right around pH of 2. That's its optimal pH. And so that's where it's going to have its most activity, right? So the rate of reaction is going to be the highest at pH of 2. That's its optimal conditions. If we take a look at some of these other enzymes, like trypsin is the green one, and then they just have most other body enzymes. We're right around here. So we're kind of in this range between 7 and 8. Okay, 7 is neutral, 8 is a little bit alkaline. So most of the enzymes in our body, in our blood, in our lower digestive system past the stomach, that's where trypsin comes in from the pancreas, their optimal pH is right around 7.5 or 8. So if we were to put trypsin or some of our other bodily enzymes in our stomach, they're going to denature. It's too acidic. Or if we put pepsin from our stomach into our cytoplasm or into our duodenum, where we have a more alkaline pH, the pepsin's not going to work. It's too alkaline. So for enzymes to work best, when we talk about pH, we want them to be in these optimal ranges. And you can see by the graphical representation, the top of that curve, that's going to be the optimal conditions. All right, we have a similar condition for temperature. The curve isn't quite so evenly distributed on both sides of the optimal temperature. In this case, we're right here at that optimal peak. But if you take a look at it, the colder temperatures, we still have some reactions, but they're pretty slow. Cold temperatures do not denature the enzyme. So cold temperatures, think that your molecules bumping around. Ooh, you're really slow, you bump in, you might get a reaction, but everybody's moving really slow. And then the temperature warms up, bonk, you get a reaction, bonk, you get a reaction, bump, 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 bump. You get a lot more reactions the faster the molecules are bouncing around. So that's why we see this slow increase in rate as we increase temperature. We're increasing that kinetic energy of the molecules bumping around. And so that increases the frequency that you're going to have the substrate bump into your enzyme. So your reaction rate's going to go up. But then if the kinetic energy is so fast, that's high temperature. Temperature is a measurement of kinetic energy. That quickness of that molecular movement could be enough to start breaking those bonds of your enzyme, right? Your primary, secondary, tertiary, quaternary structure, and you're going to lose that three-dimensional shape. So when the temperature gets too high, it's going to denature the enzyme. And we are in danger of that when we get really high fevers. So low fevers are pretty okay, like 101, 102. I'm not a doctor, so don't, always consult your doctor if you're concerned about a fever. But lower fevers are fine because our body can tolerate. We have this range, right? So we are still within a pretty decent range at temperature 104, right, Fahrenheit degrees. But once we get too much past that, we're kind of down that quick downslope where denaturing of enzymes could occur. So when we have a very high fever, like 108 or higher, our cells can have serious damage, sometimes permanent damage, especially in our brains because our neurons are very sensitive to changes in optimal conditions. So the enzymes that allow our neurons to work properly, if they become denatured, they may not be able to return to functional status, and then your neurons are permanently damaged. So like hyperthermia or fever can lead to denaturing of enzymes. On the flip side of that, hypothermia, we're not denaturing things, but things slow down so much that we cannot maintain homeostasis. Our reactions are just not happening quick enough. Okay, so that finishes up the chapter 6. So again, as you're going through this and you're like, that just seemed so random, like there's all these ideas. Do the best you can at kind of working through it, getting some ideas founded. And then once we get into the next couple chapters, you actually start seeing those topics applied, which sometimes is really helpful for getting a good understanding of what they are. So do the best you can working through this chapter 6 material, and I'll see you next time with chapter 7. Bye.